All right, it's time to extract some urease from soybeans. The very first thing you want to do is thaw your cabbage juice indicator. You can thaw it in the fridge overnight or just on the counter for a little while. And then you want to gather your materials. So that thawed cabbage juice indicator, your lab kit for urease extraction, and then also your well plate, which you may have used in the last lab. Please make sure that it is clean. Make sure you're wearing your safety glasses and you're doing this lab away from any open flames because the acetone is slightly flammable. Now you are going to extract urease from the soybean meal. So you want to first label your big tube with urease in 20% acetone and your initial state and period. You should then weigh out the correct amount of soybean meal and add it to your labeled tube. Next, you're going to locate your tube of 20% acetone. It may not be in your kit because I gave it to you to put upright in a cup when you got home. You will have a 50 ml tube with acetone in it. You want to pour the entire contents of that tube, which should be about 20 mils, into your soybean meal tube. You should then mix the soybean meal and acetone tube thoroughly by inverting for about 2 minutes. You want it to be thoroughly mixed. You can then create a hot water bath or warm water bath using warm tap water in a mug and set the tube in there for about 5 minutes. That will help to purify some of the solution. Meanwhile, you should rinse the acetone tube and relabel it as 10 milligram per milliliter urea. You then want to weigh out the correct amount of urea and add that to this tube. You should then add tap water up to the 20 ml line to the urea tube and make sure you mix it thoroughly until it all dissolves. You're now ready to set up and test whether the urease in your urease and acetone tube that you've just extracted from the soybean meal is actually active, is actually doing its job to break down urea. You should make sure to have all of your materials with you before you start, and you want to make sure to have your notebook open to the diagram you drew about how your well plate will be set up. Okay, so you want to make sure you have your cabbage juice indicator, you have your urea solution that you just made, you have your urease solution that you just extracted from the soybeans. You'll want your 1 ml syringe, some dropper pipettes, and some water. The first thing you should add to the wells is the correct amount of tap water for each well based on your notebook. And then you want to add the urea solution to the wells that need urea solution. That's 1 milliliter using your syringe. Make sure to rinse the syringe after. Next, you want to take your urease in acetone tube and make sure the sediment has settled to the bottom. And then you're going to take solution from the top of that tube and add it to each of the wells that needs urease. Make sure you've rinsed before and after using the syringe for this. Once you've added all these colorless solutions to the correct wells, you're going to add the pH indicator to give a color metric of whether or not a reaction has taken place for each particular well. You want to add a standard amount of indicator solution to each well. So if you add 10 drops to the first one, you should add 10 drops to the next one, and so on. And then you're going to observe your results. If the color is very faint to begin with, you may want to add a few more drops to each of your wells of the cabbage juice indicator, and then you may want to give them a very gentle swirl, making sure not to spill, and then wait a little bit of time to see if a reaction will take place. Then you want to place your well plate on a plain white background, so printer paper would be best, not paper towel or anything like that. And you want to label on the white paper background what is in each set of wells. So for instance, I've labeled here typing, but you should handwrite on the paper what is in each set of wells. And then you want to take a clear photograph where the color difference between the wells can be clearly seen from above. Once you are sure you have a good clear picture that's labeled and shows the colors well, you want to clean up. And you want to be sure that you wash, dry, and save your well plate, your transfer pipette, and your syringe because you will need them in future labs. You should put your tube of urease in acetone, your tube of urea, and any extra cabbage indicator into the fridge. You will need those for your next lab, so please do not get rid of them. Make sure they are clearly labeled and stored somewhere safe in your fridge.